After the baby is born, the placenta naturally separates from the uterine wall. This marks the third stage of labor. Once detached, the placenta is no longer connected to the mother's blood supply. Without continuous blood flow, placental cells rapidly begin to shut down due to ischemia, meaning a lack of oxygen and circulation. For this reason, the placenta is considered non-viable tissue shortly after delivery. However, in advanced laboratory settings, scientists can temporarily maintain placental activity. Within minutes after birth, typically within the first 20 minutes, researchers can connect the placental blood vessels to a specialized perfusion system. This system pumps an oxygen-rich, nutrient-containing fluid through the tissue, mimicking blood flow. Under these tightly controlled conditions, parts of the placenta can remain metabolically active for approximately 2 to 6 hours, allowing scientists to study placental biology, drug transfer, and oxygen exchange. By keeping the placenta alive in controlled laboratory systems, researchers can safely study how oxygen, nutrients, medications, and even viruses pass from a mother to her baby, without putting pregnant women or fetuses at risk. This research is helping scientists better understand pregnancy complications, improve drug safety, prevent infections, and develop new treatments that protect both mother and child. Placental blood and tissue are exceptionally rich sources of stem cells. These cells can be safely collected at birth and cryopreserved, stored in liquid nitrogen, for decades. While frozen, the cells remain alive in a dormant state, but they are inactive and do not function. Because placental stem cells are biologically younger, more flexible, and less specialized than adult stem cells, they have a greater ability to transform into different cell types. Today, they are already used in treatments for blood disorders, immune diseases, and certain cancers, and are actively studied for future therapies aimed at regenerating damaged tissues and treating conditions once thought to be incurable. In 2018, researchers successfully grew mini placentas, also called placenta in a dish, in laboratory conditions using real placental cells. These organoids are tiny three-dimensional clusters of cells that closely replicate the structure and behavior of a first trimester placenta. This allows scientists to explore the earliest stages of pregnancy in ways that were never possible before. Despite being only millimeters in size, these mini placentas perform many of the same essential functions, including hormone production and nutrient transfer. Because they can survive and grow in laboratory conditions for extended periods, mini placentas give researchers a safe and ethical way to study pregnancy, drug passage, and infections, without testing on pregnant women or developing babies. The placenta is no longer just something that ends with birth. By preserving and studying it outside the body, science isn't trying to replace nature. It's learning from it, and knowledge that could protect generations to come.